and welcome to Rocket Biologist, the show where we meet experts in medicine and biology. I'm your host, Dr. Adil Nasir. While recording this video, a song is playing in my head, Gravity of Love by Enigma. It just made me think that how much life on Earth loves terrestrial gravity. The romance of life with Earth's gravity carved us literally the way we are shaped. Even a single-cellular organism has mastered a trick how to sense gravity. It's called gravitaxis. A recent study has shown that even sperm cells, human sperm cells, can perform gravitaxis. Did you ever wonder that how do we know what is up, what is down? How the altered gravity environment beyond Earth will impact the living systems? And most importantly, how a single-cellular organism can sense gravity? To answer all these questions, we are talking to a legendary scientist in the field of gravitational biology and space research. My cosmic comrades, I present to you Professor Dr. Donat Peter Häder. I had a lovely time talking to him. I hope you enjoy this conversation. Hello and welcome to Rocket Biologist. We are talking to Professor Donat Peter Häder. It's great to have you on the show. And let's get started the conversation. So I would like to ask the first question. Who is Donat Peter Häder?
Well, I'm a botanist by training. I got my PhD from Nulch, who was the key figure in botany in Germany. He wrote the Allgemeine Botanik, which is the textbook for students in botany. I did my studies in Marburg and then also got my PhD there. Later on, I went to the United States for a year to East Lansing, working at the DOE. When I came back, I continued working in Marburg for a little while, and then got the call on the chair in Erlangen, where I became the main professor in botany. At the same time, I became director of the botanical garden, and later on, director of the Botanical Institute. In the beginning, I was interested in cyanobacteria, which was also the topic of my thesis in the motility and orientation with respect to light. And later on got into Euglena, and I'm sure we will be going to talk about that microorganism a little more. That sort of hooked me. There's a whole community in the world working on Euglena, which is a unicellular microorganism, which has very interesting capabilities. It can orient with respect to light, either going towards the light source or away from the light source. And it's very competent in doing gravitaxis, orientation with the gravitational field of the Earth. We try to understand how that is possible in an organism which is less than 100 micrometers long. Physicists told us, well, this is quite easy. It's a physical process where the rear end is a little more heavy than the front end, so they sort of adjust like a buoy and swim upwards. And I never thought that this is really true. So we started working on it together. Well, pretty soon we started working with DLR, with NASA, with ESA, and the Russians using their satellites, rockets, and other space hardware. And finally, we succeeded in understanding this fairly tricky process of gravity orientation. So I retired in 2009, but still keep on working with international scientists in South America, like Argentina and Brazil. I'm a visiting professor in China, in the University of Xiamen. And I'm still working with my colleagues in both Pakistan and in India. We just finished a textbook, which again is on cyanobacteria because they are focusing in this field. So we have been collaborating with this, and it just appeared with Springer. So I'm trying to keep busy and doing scientific work, writing reviews, papers, and going to conferences. I'm a director of a German photobiological society. We just had our annual symposium this year, and that was quite interesting. So I keep being busy with that. Thank you.
Well, that's a wonderful. You have hands in many pies, I have to say. So, will you please tell us about your metamorphosis from donut to Professor Dr. Häder?
Interesting way of putting it. Well, Donut is my first name. Well, after the PhD in Marburg, as I said, I became professor in Erlangen and continued there all the way to my retirement. And then I actually, in terms of my collaboration with our Brazilian friends, I was involved in their education at a university of Joinville, which is in Santa Catarina, one of the southern states in Brazil. And because of that, well, we gave courses and lectures. They gave me the honor of giving me a PhD honoris causa, which I'm kind of proud of.
How did you become interested in the field of gravitational biology and space biology?
Well, that was a kind of lengthy story. As I said, we first studied photo orientation in Euglena, trying to find out what the photoreceptor is. We did not solve that riddle, but our Japanese friends in Okazaki, they used molecular biological techniques and found out a very interesting and very rare photoreceptor. It's not a retinal, it's not a flavin, it's a fairly complicated molecule. Actually, there are two molecules. And then I got in touch with DLR, which is the German equivalent to NASA. And they are interested, of course, in gravity orientation. So we looked into the organism and found out that this is fairly interesting. They suggested, why don't we concentrate on that? The DLR has the possibility of various tools, such as rocket flights in in Sweden, northern Sweden, in Kiruna. So we designed the first experiment there, and we were capable of looking into the orientation patterns while the rocket was flying. It's a parabolic flight, and during a fairly short time of 6 to 8 minutes, you could see what happens if the organisms are exposed to microgravity. And then we continued with that, making it more and more complicated, and trying to figure out what the receptor might be. And there were some funny stories about it. Usually, when we look at our samples, they were going away from the center of the Earth, so showing negative gravitaxis. And then all of a sudden, I had a student who came and said, no, she had some recordings of the organisms doing positive gravitaxis. So first I didn't believe that, and then we continued looking into it and found out, well, that depends on the age of a culture. If you take a culture and re-inoculate it into fresh medium, in the beginning they go down, positive gravitaxis, and then later on, after maybe 2 weeks or so, they become negative gravitactic. And one of my co-worker showed you can induce that also either by using metallic inhibitors or by exposing the organisms to high visible photo or UV radiation. And by that you can invert the direction of gravity orientation. That all was an indication that, yes, this is not just a mere physical process where they happen to be oriented in the water column, but they do orient by a specific mechanism. And then we started also together with our Italian friends from Pisa, from the CNR, to look into possible inhibitors of gravity orientation. And yes, indeed, when you add certain inhibitors, certain drugs, the organisms do not know where's up or down, but go random. There was another indication that, well, this is really a sensory process, a physiological process based on a gravity sensor, a receptor. And the interesting thing is, while we were looking into the gravity orientation mechanism in this organism, we knew that nobody really knows the mechanism for gravity orientation in humans. We know a lot of interesting features, as you know, if you have too much alcohol, then your gravity orientation doesn't work too well in humans, but we do not know what the gravity receptor is in humans, the molecular receptor. I have some ideas what it could be, and I'm still waiting, but maybe it doesn't work anymore, to solve that, which would be a very interesting step forward in human physiology.
Thank you for the elaborated answer. So can you please define gravitational biology for the audiences? That what is gravitational biology?
Yeah. Well, it's simply the response of an organism to the gravitational field of the Earth in this case, but it would also work on other planets. in fact, we did some interesting experiments on the space shuttle with the American shuttle Columbia, and we had a centrifuge on board of the shuttle. So we put the organisms inside. Well, I wasn't involved in the actual flying. We had astronauts doing that, but we saw all the recordings and the experiment while it's taking place. And then they started with microgravity. The organisms went random, so they didn't know where, well, there was no up or down. Then they slightly increased step by step the acceleration by the centrifuge. And at a certain point, they started orienting. And that was at 0.4 g. So the organisms are capable of orienting with respect to gravity. And that would even work on Mars or on the moon, but not on Earth, not in microgravity. So this is probably a built-in safety. If you're capable of doing it at a fraction of gravitation of 1g, then you probably are well equipped to be very well oriented. And it's really amazing when you look at the videos in a vertical cuvette. These cells really swim straight upwards, or as I said, downwards when they're young.
That must be a breathtaking image to look at. I've been playing with that, this Euglena. So it's nice to always watch them swimming up and down. So what role does gravity play in the evolution of different life forms on Earth?
Well, obviously, most organisms are capable of sensing gravity, with the exception of maybe viruses and very small bacteria, but even other very small organisms are capable of doing it. If you consider an organism in its habitat, in an ecosystem, it has to be aware of where it is and where it's going. One thing is you need to be, if you're photosynthetic, you need to be close to the surface to get enough light intensity to survive. And the other thing is nutrition. If you have to find your food, this may be either oriented towards the surface of the water column or further down. So the organisms follow the gravity field in order to get where it's very profitable for them. And that may even change. That may even change during the day, during a 24-hour period. For instance, ciliates are capable of going up at a certain time during daytime, and they go down at nighttime, which can be due to either food availability, or it can be due to avoid predators. They know maybe that the predators are up, like fish, at a certain period of time, and they try to avoid that, which is very sensitive. And the movements in these ciliates, for instance, can be 24 meters or more, when you consider that with their length, which is also in the 100 to 100 micrometer range. So it's amazing how much these organisms are capable of doing, being oriented just by gravity.
Okay, so that's primarily you explained for the unicellular organisms. So for multicellular life forms, plants and human, what
Right.
kind of roles you can think of?
Right. Well, if you consider plants, they need to be capable of harvesting solar energy. So the best way of getting there is using gravity and growing upwards, which works pretty well for most plants. Uh, Even for mosses and ferns, they use gravity to get oriented in their life environment, and this in plants is fostered by the availability of light. When you look in a speeded up video, then you can see that the plants get oriented and fold their leaves in such a way that they uh, get them into a space where they're exposed to light. If they're, for instance, under the cover of a higher plants which grow above them, so they need to find a solution to find a space where to harvest not just enough, but the real amount of light. Too much light can also be very bad. As we know, there's what we call photoinhibition, which is a mechanism by which high light intensity, like solar light at noon, blocks photosynthesis by um, uh, uh, killing certain proteins in the complicated photosynthetic machinery, and that is being prevented by moving out of there. So when you look at plants in a fast, high-speed recording, you see that they are moving all the time to adjust their position in space, in their habitat, in the ecosystem.
And what about humans? Like how gravity is like playing role, and that will be relevant when you see future of humanity the in space. How that will matter for human.
Yeah, that is kind of interesting. There are lots of studies doing that. Well, we all walk straight upwards with our legs down and head up. But when it comes to microgravity, then things start getting complicated. That was noticed in the early days of space flight. When astronauts were exposed to microgravity in a satellite, they had to adjust. And later on, as we know, when you've seen the video sequences from the ISS, for instance, where astronauts live for half a year or more, then this is complicated. They try to adjust in their environment. But the other thing is that there are lots of problems involved. For instance, you have a gradient in your body. And when there's microgravity, for instance, your blood is distributed differently in your body. So they have to get adjusted to that. You don't need your muscle strength to keep upwards. And all the astronauts feel that they have reduction in muscle volume and try to counteract that by gymnastic activity on the ISS, for instance, and even in the short-term microgravity, you feel it. I myself was involved in zero-G experiment on a flight. They do these parabolic flights from either Cologne or from southern France, from Bordeaux.
Yeah.
And there you, the pilot takes the plane up to a certain height, then it sort of drops down during like 6 seconds, then he stops it again, flies up again, and there's a oscillation of 20 such movements, which exposes you from 1G on a normal flight to 0G when it's going down to 2G when it's pulling up. So you notice that when you're exposed to that sequence of events, which makes it a little complicated. Well, we do that, of course, when we try to do experiments with our organisms. And at that time, you have to sort of adjust to that strange change in gravity exposure while doing your work on the experiment.
Yeah, that was fun. If you remember, this was our first campaign. I was also part of that same campaign. It was also first time for me to take parabolic flight. And how was the experience, by the way? Did you enjoy it?
Yeah, kind of. During the first time I did that, it was very novel for me, since I've never been on a rocket flight or on the ISS. So I had to get adjusted to that, but that was fairly quickly. So when you knew what you're doing and how to hold tight when when you're under microgrid G, not to be bumped around in the plane, and then once you got adjusted to that, that was kind of fun and interesting. You had, like, as I said, 20 parabolas. Then there was some time interval to get adjusted to 1G, and then they repeated the whole thing again. So it was like a few hours in the plane when you did that. And the personnel there took care that you didn't have problems. As you know, the astronauts, when they go to the space station or on a prolonged shuttle flight, they all got nausea. So during the first day, there's no real task. They just had to be adapted to the new situation. And the personnel on the 0G flight, the parabolic flight, gave us medication to avoid that disease, which you also might get on a car or on aeroplane flights. Yeah, today they give you an injection which is more powerful than just swallowing a few pills.
Well, that's a wonderful story. I would like that you'd mention about your organism of interest that you worked. Your career stands on that major contribution to gravity sensing mechanism. Can you formally introduce us to your organism of interest, what it is, and why you chose it?
Euglena is a flagellate. And the first interesting question is, when you look at it, sometimes it's even used in schools for microscopy classes in biology. And then the first question is that a plant or is it an animal? It is motile, okay. Plants usually don't swim around actively, but they are motile, but much slower. So Euglena swims. It has one flagellum inserted at the front end. In fact, there are two flagella. One is very short and stays inside the body. The other one is used to power the organism. And it goes on a helical path. So it swims helically, which we found out later on is important for the mechanism of orientation, because you get a modulation in the system, in the signal. Well, and then it has chlorophyll, it has chloroplasts, and that is kind of interesting. So you could say it's a plant. But there are forms, various other euglenas, there are many species, and some of the species do not have chlorophyll, no chloroplasts, and they feed just heterotrophically. Okay, so the current idea is that a heterotrophic organism had taken up chloroplasts to serve as symbionts. And that happens also in higher plants, for instance, which took up, via mosses, ferns, and other organisms in between, took up cyanobacteria, converted them rather than feeding on them, but using them as slaves, as symbionts, to produce the organic material for them. And the next step is when you look carefully in an electron microscope, you see that the organism, that the chloroplasts in Euglena have three membranes. In higher plants, they have two membranes. One is from the cyanobacterium, which was taken up. The other membrane, is built from the plant organism. But in Euglena, obviously, they took not up cyanobacteria, but already eukaryotic organisms, which had already these two membranes. So Euglena built a third membrane. So we call that a third symbiosis, which is kind of interesting. So whatever you want to call it, a plant or an animal, doesn't matter really. It's a unicellular organism which may or may not use chlorophyll to do photosynthesis at the same time. You can even use chemicals to wipe out the chloroplasts within the cell and then feed them heterotrophically by sugar or other organic material, and they can quite easily survive. And when we talk about survival, the other interesting point is that these organisms are basically immortal. They do not die. They simply divide again and again and again until they come either eaten or come into a very bad physiological situation. But theoretically, they could survive forever.
So you're referring to the symmetrical duplication, that if it's not asymmetrical, so they have chances to keep on going and no chances of aging, they show. That's one of the reasons that Euglena has been used for studying how aging is working in other life forms. Is that correct?
Yeah, I'm sure. That is an interesting and important aspect, and theoretically or ethically quite interesting. These cells, Euglena, does not have sexuality. We've never seen any copulation or exchange of genetic material. Even bacteria have that. As we know by now, they can exchange genetic material, but Euglena doesn't use that, but rather has the capability of dividing. That has important implications on the genetics and the evolution of the cells, how do they do any evolution, change in the DNA content, change in genes, if there's no genetic exchange with other cells.
That's fascinating. So, can you give us the real life examples that where Euglena is showing the gravity sensing mechanism, where one can see if Euglena is there, like how
Yeah.
it works in nature?
Yeah, well, when you walk into the countryside, these cells are almost everywhere. They have many habitats, even small puddles or lakes. You can find them, but as I said, they're very small. You can't see them with your naked eye. You see only if there are large populations of those. And one of the first time when that was seen was in the wastewaters of a sugar mill, where they used the organic material, so the whole thing looked green on the surface. And when you scoop that up, take it to a microscope, then you find the cells in there. And there's another one, which is Euglena sanguinea, which is red because of a certain carotenoid which it produces. And then all of a sudden you see red puddles, and in areas where there's enough organic material, then you have the red puddles, especially during daytime when there's sufficient light. They all accumulate at the surface, and during nighttime they go down because there is no light energy, so it's not necessary to be there and be exposed to potential predators. That is an interesting organism. We never, it has been known for many decades by now, but we never were capable of cultivating this Euglena sanguinea, the blood Euglena, if you want to translate that. For some reason, something seems to be missing. You can keep the culture for a while, but it will not grow and divide and enlarge the population. Strange enough.
Okay, so you already touched upon that question, but let's say we can combine these. So, what is the evolutionary advantage that gravity sensing mechanism have for Euglena? There is, as you mentioned, there is a light source as well. Euglena also sends light, but are there other stimuli that Euglena utilize to orient itself in a certain way in the nature?
Yeah, definitely. When you look at these organisms, as more or less all organisms, including microorganisms or multicellular organisms, they respond to a multitude of external stimuli. Well, we have discussed light and gravity, which are strong stimuli to guide you in a favorable habitat and surrounding. But these cells also respond to oxygen. So imagine you have to these cells, when they're doing heterotrophic feeding, they need oxygen, and then so they orient with respect to the oxygen concentration in their surrounding, which is called aerotaxis, which is also evolutionary kind of interesting. When you imagine the first organisms appeared on Earth about 3.5 billion years ago. And the first organisms which did a photosynthesis, like higher plants, more or less, were cyanobacteria. And they were the first to produce oxygen. There was no oxygen in the atmosphere. Nitrogen, CO2, other gases, but no oxygen. And then it took them about 1 billion years to produce oxygen, which did not go into the atmosphere because it was used to oxidize iron, which was in the Earth crust. So 1 billion years, the situation in the atmosphere was the same. And only when all the iron basically was oxidized, rusted, then they started building up oxygen in the atmosphere, which we find today up to about 20%. But on the other hand, you have to imagine oxygen is poisonous. It's a toxin. And many of the other organisms at that time, bacteria, which also did photosynthesis but did not produce oxygen, had a big problem with the rising oxygen in the atmosphere. That's why we see these organisms, these bacteria, only in anaerobic places where there's a very low concentration of oxygen. So even for humans, oxygen is toxic. When you're a diver and go to a certain depth and you breathe fast, as beginners do, you are toxified by the oxygen and get like dizzy, as if you've drank too much alcohol. And you have to be careful about the high oxygen concentration which builds up in your blood. So this applies probably to all the other organisms. They have to find an area where the oxygen is kind of positive for them, the oxygen concentration. Other stimuli in the surrounding could be magnetism. Some organisms respond to the magnetic field of the Earth. In Euglena, I tried a lot of times, but couldn't find that. But on the other hand, they respond to an electrical field. Well, you may know that in the environment there are low voltage electrical fields, and we are not sure what the profit is for a cell like Euglena or other organisms, but they do respond to the electrical field, either going towards the anode or the cathode, and get oriented within that field.
So how do they sense that fields, like magnetic field and electric field? Do they have sensors for that?
We don't know. There's a guy in the United States who was studying the aerotaxis, the oxygen dependence, but hasn't found yet a sensor. Probably they, well, by whatever mechanism, they sense the concentration. And what they usually do is they go up or down a gradient. So they do not measure exactly and tell you, well, this is 20% oxygen, but they find out, okay, it's increasing or it's decreasing, which is not only true for oxygen, but also for other chemicals. For instance, it's important if you take a strain which is not photosynthetic, that you get enough organic material to feed on. So they follow gradients of sugars, other carbohydrates for getting sufficient amount of food. Again, we don't know how they do that. And in many cases, it's not really known. What we know in Euglena is the fact that what the mechanism of sensing is and the mechanism for photosensing, but not the other responses to environmental stimuli.
My cosmic comrades, we will continue our intergalactic conversation in the next episode. Until then, stay curious and stay connected as we are just beginning our odyssey into the heart of the universe.